out 1660 to be amplified.
Well, you can say guaranteed, but like guys are
Okay. Okay, 2 minutes. Please come here. Okay. Okay. All right. Yeah. Okay.
Typical. Can I open our laptop? Is that right? Yeah, of course.
Hello. Just grab
More seat?
Do you want, I guess I-
How many?
Yeah.
Oh, that's right.
Yeah. We need to move a little bit over there. Gentlemen.
Thanks.
Just
No worries.
Just because we're doing it in the video.
Whew, the lights again.
Okay, so I think we'll get started. So first of all, thank you, people in the room, for coming. Also, people online. We've got people on the conference platform, but also on YouTube as well. So I hope you're all looking forward to hearing from some of the kind of most exciting research and science and content from the conference here today. This is an opportunity for you all to hear directly from them and ask kind of questions as you want later on. In terms of the format, so we'll start with an opening remark from Christoph, and then followed by 5 minutes from each of the panelists. After that, there'll be a Q&A session with journalists, and please do either use the QR code there to ask your questions, ask them in person. On YouTube, please put your comments onto the channel, and we will be picking them up and asking them as well. So there's plenty of opportunities to engage with the panelists. Before we start, I'd like to ask Dr. Christoph Langer, who is the Secretary General of the Union, just to say a few words.
Yeah, thank you very much. On behalf of the union, it's a great pleasure to welcome everyone and to extend my also congratulations to those who have been selected to sit here as representing the highlights of the conference. It's an enormous pleasure to be back in business after 3 years of COVID and to meet in person. I see many happy faces walking around the conference, and many people are really grateful for having the opportunity to share their newest developments in research with others and to meet colleagues that they haven't seen for a long time. We're also living at very exciting times. Never before so many people have been affected by tuberculosis in the world during 1 year, as reported by the WHO last year. And therefore it's a great pleasure to introduce today highlights from the conference and the latest developments in terms of TB prevention. Professor Anneke Hesseling from Cape Town will report on a clinical trial on TB prevention in children. We have 2 very exciting presentations on tuberculosis diagnosis. One is on a little device, battery-driven, that can be used in the field to accurately diagnose TB with a very novel technology. And then there is an exciting technology with animals, giant African pouch rats, which are able to sniff TB bacteria from sputum and able to guide the diagnosis of TB in resource-limited settings. And last but not least, we will have one presentation of a novel drug that is currently in phase 1 of clinical development, which shows very promising results. It's a drug that is similar to bedaquiline, but yet has better properties in phase 1 clinical trial, and which may have the potential actually to overcome also bedaquiline resistance in clinical practice. Also seems to have a very good safety profile. And with this, I hand over back to Alex to introduce the individual speakers.
Yes, so if we could start with you, Anneke, and we'll go from your left.
Thank you so much. It seems like the theme is of mice and men. So we'll move from rats to children. So we're really pleased today to have been able to share the main results of the TB CHAMP trial, which was really looking at prevention of drug-resistant TB in children who've been exposed to multi-drug-resistant TB in the household. And as a bit of background, multi-drug-resistant TB in children in the pediatric population is probably the most neglected of all the populations affected by MDR-TB. Fewer than 20% are actually diagnosed and treated. And so to find more cases and prevent more cases is really, really critical. The treatment of MDR-TB in children is still quite complex, lengthy, it still has a lot of side effects. Kids have not yet seen all the benefits of the therapeutic advances seen in adults. So prevention is really key. And the TB CHAMP trial was really a phase 3 efficacy trial looking at levofloxacin to prevent new cases of TB in children, and also looking at the safety of levofloxacin in preventing TB in young household contacts. So this is a study that was conducted across 5 sites in South Africa. And we also were really severely affected by COVID, and being able to implement and complete the study during COVID was really challenging, and I'm very grateful that we could complete that. In essence, we showed that levofloxacin, compared to placebo, reduced the risk of developing TB in young children by 56%. So that's an almost 60% reduction in developing TB. And very importantly, for this class of drug, we showed that levofloxacin was extremely safe and very well tolerated in kids. In addition, we also looked at whether the strategy was actually acceptable to families and households, and it was. And finally, in data that will be shared formally later today during the meeting, we've looked at whether the strategy is actually something that could be cost effective and affordable, and we show that the strategy is not only cost effective, but it actually is cost saving. So in essence, we have, for the first time ever, a randomized controlled trial to provide data on how to prevent TB in kids exposed to MDR-TB. What is really exciting is that an adult trial was conducted in Vietnam at the same time, called VQUIN, and the 2 trial teams collaborated early on to combine our data, harmonize endpoints, so that we could actually jointly share and present data across the age, basically from age 0 to age 99. And we showed actually across the age spectrum that from kids to adults to old adults, we could actually effectively reduce the risk of TB by 60%. So I think the value of collaboration and data sharing has been really, really clear, and it's very compelling. So we're very grateful to our funders and our collaborators for supporting this trial and making it possible. Thanks.
Thank you, Annika. Maria, over to you. Yeah.
Good afternoon. Thank you very much for the opportunity to join you today. What I'd like to talk to you about is our new phase 2 study that we've just initiated on a novel compound, as it was mentioned, TBAJ876. So before I get into the details of the clinical trial, I'd like to say a few words on this novel compound. It belongs to the class of the quinolines, which is the same class as bedaquiline, cornerstone in the treatment of tuberculosis that was approved for use over a decade ago, I think. And this novel drug is very promising because it's much more potent than bedaquiline, tenfold more potent. It retains activity against the most frequent form of resistance against bedaquiline. So again, the potential for a treatment that could be universal for tuberculosis, and also has the potential for being safer, especially no QT prolongation liability, which will facilitate monitoring of patients. So because of all of these attributes of TBAJ876, we initiated a phase 2 study, also very important and attractive because it's a study that's combining a number of different aspects of study design from phase 2A, phase 2B, and phase 2C. All of these trying to accelerate the development of this compound that could be very important in the armamentarium of tuberculosis therapies. So what is this study? It's a clinical trial that's going to enroll around 300 patients who are drug sensitive and in 5 countries, South Africa, Georgia, Tanzania, Uganda, and Philippines. And these patients will be randomized to 5 arms, 3 of which will contain TBAJ876 in a dose-ranging format in combination with 2 other compounds, linezolid and pretomidine. And so that's, I guess, one of the advantages of this study. We're going to be able to do dose ranging for this novel compound administered on a backbone that's well characterized, the combination of pretomidine and linezolid, in this way offering more benefit to patients than they would have had if we would have done this in monotherapy, in a monotherapy study. In addition, we're going to have an arm of BPAL, so that is bedaquiline, the compound I was talking to you about, the first class, quinoline, also in combination with pretomidine and linezolid, which will allow, in a certain way, the comparison of TBAJ876 at the 3 different doses and bedaquiline to confirm the higher potency that this compound has and also the improved safety. And finally, there's going to be a control group. The primary endpoint of this study is going to be at week 8. We're going to be measuring across the arms time to negative sputum conversion. But the study doesn't end there. Patients will continue treatment to complete a full course of therapy. Most of the patients will be treated for a total of 26 weeks. In the arms that are going to be receiving TBAJ876 in combination with pretomidine and linezolid, based on their response at week 8, will allow patients to complete therapy after a total of 15 weeks of treatment, already exploring shorter treatment durations, which is going to be a very important aspect of this design to guide us in our phase 3 study that's going to follow. And the very last thing I'd like to say about the study is that, as I said, there is a BPAL arm in which patients will be receiving the standard treatment, now indicated in patients with resistant tuberculosis, but this is a drug-sensitive study. And the objective of that is to learn of the safety, tolerability in this patient population, but also the efficacy, so that we can compare with our knowledge on drug-resistant tuberculosis. And this regimen could actually become the first universal treatment for tuberculosis, to be replaced maybe by a better constructed regimen, including TBH-876 later on. The study is ongoing. There's 2 out of the 5 countries already activated. We have recruited already a number of patients. We expect all sites to be activated by the end of the year. Thank you.
Thank you, Maria. Tony, over to you.
Yes. All right. So this is Tony Hu from Tulane University at in the USA. I really appreciate Chris and others' leadership to include me as an engineer and to join the union conference, because today, all the pathogens become more intelligent. It really needs of a multidisciplinary effort from the engineer, chemist, and also the clinicians to focus on the one aspect of the disease and to address the critical problem for the clinic. So for us, our team has been focused on the identifying and quantifying the blood-based biomarkers for tuberculosis diagnosis, and starting from the antigen peptide to extracellular vesicle, and also recently the cell-free DNA, and we have been making the 10 years of effort on this, and so. This is actually for the detection of cell-free DNA, actually starting from the pandemic time. And when the pandemic just started, and clinicians urgently need a more sensitive detection assay to identify the real patients, especially at the hospital. We have so many patients under the cancer immunotherapy. So, but COVID really cannot be detected accurately. Then we're just thinking, well, if the PCR doesn't work, is there any other option for us? And then we actually select the CRISPR. But at that time, the CRISPR technology, they can only detect the high abundance biomarker and with the poor specificity. Then we actually did some improvement, and by including the 2-step of amplification, and also building up the high throughput screening method for COVID. And that sense, the limit of detection can be down to 0.05 copy number per microliter. And after pandemic, we think, well, how we can serve this technology for tuberculosis. Is there any chance we can capture the circulating cell-free DNA from the blood, and then we can then better serve the patient with extrapulmonary TB, HIV infection, and also pediatric patients. Then the same thing, we also using the COVID platform to detect the cell-free DNA, and we collaborate with the Baylor the College of Medicine team, and also the University of Washington team, and running this assay in the 2 different cohorts. One is for adult, the mixture of adult and children. So it's more like the validation, pre-clinical validation study, and the sensitivity in the adult is like 92%, and the specificity is 94%. And in children is 83% sensitivity and 96% specificity. But it's a small group. But next, we actually work with the Kenya Institute and running and challenging this assay in the most difficult cohort, which is HIV and TB co-infected children. So this is like 150 patients, and including the 13 confirmed TB and 63 unconfirmed TB. So we capture all patients with confirmed TB, but in the unconfirmed TB, that's actually missed by the current gold standard. We captured 55 of them. So then after this, and we're thinking, well, how to further improve this and to serve the patient living in the resource-limited area. Then my graduate student just started working on this fully point-of-care stuff, and we call it a lab in a tube. So we incorporate all the CRISPR reagent in this paper strip, and here we're also using the 2 primers. One is reverse, the other one is forward, to specifically capture or lock down the target we're trying to detect, and then this spring the spring can operate this cap and allow the CRISPR reagent to fully interact with the DNA extracted from the bottom. Then afterwards, we also have this small device and put in this hole for pre-amplification. After 15 minutes, you take out and put in another site. That's a detection site. So then the fluorescence, the signal will showing up on this screen. So then we also running this validate this assay with another pediatric cohort from Dominican Republic, and is showing the nice sensitivity and the specificity. And of course, we're thinking that's what Chris asked me before the meeting, and we're also trying to expand this platform to cover more drug-resistant fragment, and waiting for the next validation. So yeah, this is the platform technology. It's not only for tuberculosis, we're also running this for the swab test for pneumonia, pediatric pneumonia, and also the MicAvia complex detection. So if you're interested, feel free to stop by. Thank you.
Thank you, Tony. Over to you, Tibo.
Okay, no, thanks Alex and the Union for inviting me for this opportunity. I think research innovation being one of the pillar of in TB strategy. I'm glad to see the diversity here now. From this device, I'm taking you to the rats now. Okay, my name is Tafarra Gizou, and I'm head of TB at Apopo. Apopo is a Belgium-based NGO working on landmine detection using animals as well as rats. So our headquarters is in Tanzania, and we operate in Tanzania, Ethiopia, and Mozambique. So TB detection using animals, a family of Chrysetomi, has been more than a decade in Apopo, and we have been trying to help in finding the missed TB cases in these high burden countries. So so far we have evaluated almost close to 1 million samples from over 500 patients, and the rats were able to find about 28,000 new missed cases, which otherwise were not found by the routine program. So what brought me to this table is this thing has been going on for a while, but this time around we aimed to evaluate what impact they are making comparing the routine system versus rats, and we wanted to also compare between children and adults. Also we want to compare between the gradient of smear microbacterium positivity. So we found an interesting finding. I think our usual finding, we almost found about 4,000 TB cases in 69 health facilities in Tanzania after screening over 35,000 patients, and then of which over 50% actually were picked by the rats. So that was contribution by the rats, which otherwise would have been missed, as I said. So further, we went ahead and analyzed, comparing, as I said, children and versus adult, So we found that they were actually able to find 2 times, it was 2 times more likely to find TB in children by the rats compared to smear or even gene expert for that matter. And again, we want to go ahead, whether, between the routine system and the Apopo rats, whether there is a difference in picking TB among the bacteriological positive gradients. And we found actually when the bacterial load is less, the rats were able to find more, actually 6 times more than actually the microscope or even gene expert. And that was very exciting even to us, so that is what makes, what I'm talking in new actually. Otherwise, TB detection by TB is not as such a new thing. Now we went ahead again, even among TB found by the rats between adult and children, we also wanted to compare that. Again, we found they were able to pick 2 times more in children compared to adults. So obviously children need all the time advocate, I guess. And yeah, we are excited that children now could have a little bit more help from the rats. And rats and children now, we move with it, I guess, in the wallet. So our main aim in Apopo is not necessarily to commercialize them, but they can make social impact by finding more TB cases in less accessible areas, in high burden countries. For example, these 3 countries, as I said, and there are more, contributing more than 100 TB,000 per year. Those are the countries that we want to move on and help. And in fact, India is also another area where, over 2.5 million TB cases are coming from. I know the technology is expanding everywhere, but again, they are not accessible to all, in different countries. So we really want to help, by finding more TB cases using our rats. Feel free to approach us. We are breeding them. We are not getting them from the bush. So we manufacture them and we habituate them. So they all, people. And then they are very friendly. So they are not our enemies, but they are our friends. So especially for African problem, these are African solutions that we want to present. So thank you for your time.
Thank you.
Thank you. My name is Zahid Islam. I work with the Alliance for Public Health in Ukraine. So I'm just trying to reflect perhaps, listening to quite a diverse research coming up and how that actually reflects to the community perspective, how we can, link with your research that you're doing and translate that into sort of, benefit for the community. So, these are very interesting, actually, looking at your tube and the rat and also the pediatrics and the new drugs. If it is, we can have one drug which is going to be able to treat, all across treatment. I think that is for us as looking at the community. I think that would be a sort of a magic bullet that we could work with, right? So for example, if we are looking at Hep C at the moment, we have this DAA, which is actually giving a lot of hopes to people. And for TB, we are waiting for long years to have something like that. So we really hope that something comes up and it will be really helpful for the community. And I just want to share maybe also in terms of context for a situation where it's not in a stable situation like the war situation and also disaster situation where reaching to people for tuberculosis when people are displaced, people are, is extremely difficult. And having something more like point of care diagnostic systems which can detect TB at the center where the people are, that is extremely important. And in Ukraine, for example, we do active case finding, going door to door to the patients. And that already gives us, nearly 2 times higher detection rate than just doing the regular TB case finding. And so I would say thank you to the research, but I think we also need to socialize those research within the community so that not actually at the back end of the research, but at the beginning of the research so that we could actually then, having those product or anything we bring out is not going to be really so difficult to accept by the community. And I would also add perhaps one more thing about tuberculosis is a social disease, as we all know. And at the end of the disease, when we are putting people on treatment and everything, I think we need to make sure that people receive patient-centered care. So even designing your programs, your research and everything, I would urge that everyone to kind of think that in your mind and keep that. So yeah, thank you.
Yeah, we'd like to congratulate you for being selected with your work, and also like to stress that the World Conference on Lung Health is probably the best forum to present such innovations in the field of tuberculosis. We've heard today that in the absence of a vaccine that is able to prevent TB, preventive therapy is the best option, and we saw a very innovative trial today presented on the use of levofloxacin to prevent very effectively tuberculosis in children. We have seen that there is a novel device coming up with a battery-driven little tool that can be held in the hand and can be used as a point-of-care test with a novel technology, CRISPR, for which a Nobel Prize in Medicine was given not many years ago, that will be able to detect tuberculosis in the field in adults and children with a very low price. We have seen innovative approaches with giant African pouch huge rats, Apopo, which are not only able to detect landmines and made a huge difference for people in areas who are affected by landmines by using these rats to clear them, but also they have the ability to sniff TB bacteria out of sputum, and as a first, also like a point of care test, to make a diagnosis with much larger volumes of samples being processed per day as it is possible with sputum smear microscopy or with PCR tests like GeneXpert, and which much less investments. The rats cannot sniff antibiotic resistance yet, which they still need to learn, although this will be difficult, but it's a very, very good point of entry test to be used in resource-limited settings. And we also heard very exciting news of a novel drug, a novel diaryl quinoline that is called TBAS876, which has the potential to replace the yet best drug that we have in the history of the treatment of tuberculosis, bedaquiline, with even a better efficacy and with at least the same safety profile. So this is very exciting. The union will, how did in the past, does currently, and will also in the future, be hopefully the forum for colleagues like you who are presenting the cutting-edge research and who will be discussing these innovations with others with the aim to improve the life of children and adults affected by this disease.
Thank you, Christoph. So we'd like to open up to the floor and also online if there's any questions that anyone has. Christoph, do you want to ask any questions? I'll check online now.
Yeah, let's start with you, Tony. Your device looks really very, very innovative and promising. When do you think will it be ready for being marketed?
Well, I'm very happy you asked this question. And we right now have the startup company called Intelligent Genome, located in Houston, Texas, and they are running the production. And also already filed FDA and got feedback from FDA. So they're moving fast because this is really the rapid test, urgent need. FDA totally understand this. So we're fixing their concern now. And then after they approve the clinical validation, and we will start. So typically for TB diagnosis, it will take 2 years to finish the validation and the clinical trials. So that's actually our target after 2 years. But on the market, you mean on the human being market, that's 2 years. But right now the company already start running this with the USDA, because some cow also have the microbovins, and they can detect it too. And also for the non-human primate centers, there's a lot of primate center. Now the T-spot is the only way they can detect the TB, but still they need to identify the monkey with active TB, so this one can help. Talking about the price, right now it's in the research and the fabricating cost for tube is $1.5. But if we move to the massive fabrication, it could be down to the $1. And the box is currently is $100 with all the part purchased from Amazon. But I believe the manufacturer will definitely lower this too.
Yeah, that will put those companies that are currently established in the market also under pressure, which is also good. Maria, the TB Alliance has made the difference in drug trials and providing novel treatment. Like your next TB trial 2020 has been a total game changer for patients affected by drug-resistant tuberculosis, showing that much shorter regimens and also in the end better tolerable regimens are possible. When I started off as a physician, I observed the AIDS epidemic and the emergence of possibilities to treat patients affected by HIV infection with antiretroviral therapies, but they had a handful of pills. Mm-hmm. About 20 years later, I saw the same in patients with multi-drug resistant TB with a handful of pills treated and sometimes very painful injections. Are we going to experience a 1 pill per day anti-TB treatment?
And that is our objective. You know, it's so thank you very much for your comment about TB Alliance. We are very excited and proud about the success with BPAL. And our intent was to try to have a fixed dose combination for that regimen already. In terms of how do we select compounds that advance in development, because we do have a pipeline, we do that by considering all of the attributes, including the one that's very important in terms of, is it a candidate for a fixed dose combination? Can we actually put it together into the one pill a day? So hopefully.
Thank you very much. And Tefera, on your rats, it's difficult to standardize a living being. So what is, could you inform us about the sensitivity and specificity of their performance to diagnose tuberculosis from sputum specimens? And they must undergo a training phase. So how long are your rats trained to be used for diagnosis, and how long do they live?
Yeah, no, thanks for the question. I passed that actually accidentally, so glad you have asked me this question. Normally they live up to on average of 8 years. They are not ordinary rats, by the way. They are called giant rats, as the name implies. So they weigh about 1.5 kilograms. They are not like house rats. And we breed them in Tanzania, and then normally it takes about 9 months, you know, to really put them on the operation. So since, you know, after 4-8 weeks, you know, they go into habituation and different kind of training. So they can serve up to 7 year, 8 year, you know, once, you know, they are up and running. So we do also internally, by the way, accredit them. We don't let them run, you know, anyhow. They have their own internal accreditation process. If they don't perform, we change them. We take them out of our system. About the sensitivity, I think Apopo has done over 20 publications on this. You can find it on the online or on the website. But there was one meta-analysis done, published in 2021 by a guy called, his name is what? I think Kanan is a meta-analysis. So from that meta-analysis, basically he used our own publication. Unfortunately, that's a limitation of 7 studies. So the sensitivity was about, you know, 18.81% and above, and the specificity was 73% and above. So we are working, you know, how they perform compared to GeneXpert. So far they were being compared with smear, where they perform over 40 to 50% over and above what the routine program is giving. We have seen some signs that they could do about 30% over and above even GeneXpert. So this time around, we are looking for fund actually to do an inferiority study with the GeneXpert. So
how many samples does one animal evaluate per day?
It's amazing, you know, they could do actually 100 samples in 20 minutes. So, yeah.
Thank you very much. If you want to learn more about the Apopo rats, you could go to YouTube. They have very interesting videos showing how the rats are trained, not only for landmine detection, but also for TB detection from sputum specimen. You see how they get the banana after a click if there is a positive sample, so that they learn actually how to sniff. It's really exciting, and this is something I would call it out of the box.
Thank you.
So,
finally, Sahibu,
sorry, Christoph. So we've had
just to wrap up very briefly, you are working in a country that is currently under siege at very difficult circumstances, and still you do active case finding. Can you tell us about the challenges very briefly on performing TB fieldwork in the Ukraine presently?
Thank you. I think so. Active case finding difficulties is that, you know, people are displaced. So people used to live in one area, but now they are actually everywhere in the country. So in order to find them, trace them, and also actually looking for these people, what kind of disease they have, and especially tuberculosis. And we also have like a specific group which are also traditionally Roma population, which are moving one place to another place, and it's very difficult to actually get to them. And as we work mostly with the people living with HIV, they are highly stigmatized as well. Even, for example, you have a displaced center, And if you are with tuberculosis, people injecting drugs, there's a different way of treating them. So they're not treated. So we have to actually struggle to kind of see how to provide them services and get to them to, actually do the screening and everything. So these are actually, it's not too many of these things are really new challenges, but it is existing challenges. But these challenges actually accelerated our case finding, difficulties higher. So, but we have field workers working, NGO people, and also civil society, community workers working in the field. They're coming from the same community, so that makes it a bit of easier and accessible for us to go there. So, yeah.
Yeah, thank you very much, Alex.
Sorry, yes. So we've got a few questions online. Maria, why is it important not just to have a single pill, but also shorter regimens?
I'm sorry, can you repeat the question?
Sorry. So why is it important also to have shorter regimens as well as just a single pill?
I think it's because we would certainly help patients to adhere to treatment if they take only one pill compared to what they used to many in the past, still a few, that facilitate compliance with the treatment. Shorter treatment duration is a similar approach. Patients will be much more inclined to accept and complete treatment that is short, and that will also liberate the infrastructure of the hospitals and settings that are caring for patients. Once the patient has finished treatment, will enter follow-up, but it's not the same monitoring. I mean, of course, as I mentioned earlier, safety of treatment is very important. The safer they are, you might not then require intense monitoring, and patients might actually be able to actually be contacted by phone in the future. I'm just imagining how things could get. So all of these components are important. It's not just the one pill or a short treatment. It has to be safe, has to be very efficient, and ideally it has to be universal.
Thank you. And Zahed, could you come in on that and your perspective?
Well, I mean, I think we did have some consultation on this, but I think basically I would agree. I mean, in a sense also it not only depends on the length, but also how you take the pills, right? I mean, would you take it once a day or 3 times a week, for example, right? So I think if you would ask me, I would say once a day sounds fine to me because I can put it as a rhythm, right? And if you have to take pills, 3 times a week, I need to think, plan, a schedule. That's a lot of effort. Yes. So every day taking pills would be much more easier. So these kind of things actually consideration are. That's why I was initially saying that at the beginning of the research, if we also engage the community, I think we can get this kind of information or feelings from the community embedded into the research. Yeah.
Anneke, what are the priorities to improve the treatment of tuberculosis in children?
Yeah, thanks for that, Christoph. I think coming back to the community, I think it's important firstly that communities know about TB in children and that can be prevented, diagnosed and treated, and that they can be very well treated, whether it's drug-susceptible or drug-resistant TB. I would say the biggest priority for treatment is finding the missing cases. Yeah. You can treat very effectively all kinds of TB. Maybe the most devastating forms of TB, like TB meningitis, is the one exception. We have a lot of post-TB morbidity and you have high mortality. But in general, children, even with XDR TB, do extremely well once they've accessed treatment. The issue is actually getting them diagnosed. And so we have the largest gap in the children at the highest risk of developing TB, which is kids below the age of 5. And for MDR TB, as I said earlier, fewer than 20% of kids with MDR TB are actually accessing treatment. So for me, treatment follows diagnosis, actually strengthening healthcare services, making communities more aware, and creating demand for kids accessing diagnosis, preventive treatment, and appropriate treatment. That's actually where it starts. So tools are amazing, but we actually need to have strong, effective healthcare services and knowledgeable, empowered communities. The TB CHAMP trial actually was a community-based trial, household-based. Before we did the study, we actually went to the local communities and asked them how would they feel about such a study, how should we be doing it, and then during the trial we engage with the community, and then are actively engaging with community and participants to be able to translate these findings. And so I completely agree that you can't just bring tools and strategies to a community, you actually need to understand what they need and what they think. But for me, the bottom line is we can have amazing therapeutic advances. Kids need to be included in clinical research more rapidly. They have been excluded from any benefits of adult proven therapeutic new regimens. So that is one way in which we can accelerate access, but that access will only happen once they actually have received a diagnosis. And in most children, that will not be a lab-confirmed diagnosis, and we need to be comfortable with that. That's a clinical diagnosis, or other amazing tools like rats and magic boxes.
That echoes very much my experience here from the conference. We have so many innovations going on, but we also have problems in getting what we would say the horsepowers on the ground. We should not talk only about R&D, research and development, but we should talk about RD&I, research, development and implementation. We need to overcome our implementation gaps to really get the new boxes to where they are needed most. If I understand correctly, most of the children who die from TB in the world have never had a diagnosis or never had treatment, and that needs to change.
Annika, we've just been asked if you could provide more information on the cost-saving element of the trial.
Yes, we'll actually be presenting data tonight. So I'm not going to steal the thunder, but essentially the once daily levofloxacin regimen was highly cost effective, and it saved a considerable amount of money. It saved a considerable number of children developing TB, MDR TB, and also TB related death in children. So, and we're looking at the costing from a societal perspective as well from a healthcare system perspective. Many preventive strategies are cost effective inherently, but having it be cost saving is really what we're looking for. So we'll leave that space for some updates later today.
Okay, thank you. And Tefera, we've had a couple more. So they've been asking in terms of how scalable is the approach? And is there any possibility of adapting this into some sort of technology?
Yes, let me start from the second one. In fact, right now we are doing a collaborative study with the University of Manchester where, we could test other samples other than sputum. For example, urine, sebum, breast, and the like. So we did get some interesting findings in that one. We could find TB, by smelling sebum, for example. So that is the one, that we are heading, in terms of something like this, with a device, to scale it up at later stage. But from the other aspect of it, what we are planning to do is we can expand at any given time into a context where, there is a volume of sample could be run, in a day. I think, as already mentioned, the throughput is very high. They can, for example, process 400 sample in an hour, for example. So you can imagine now where there is high prevalence of TB, and then if you apply this thing, it can work. So the context defines, our scaling. We don't want to bring Europe rat diagnosis here. The system works well, things like that. But the less accessible places, anywhere in the world, at any given place, we can go and expand it. So, yeah.
Do you have more, Alex?
Yes, one more. So, Annika, this is for you. For children, extrapulmonary TB is a huge concern. What point of care test works well for children in terms of sampling? Is there any progress in this area?
So I think the most common form of extrapulmonary TB in kids is peripheral lymph node adenitis, which is actually not a severe disease, and they're often actually, quite easy to diagnose by doing a fine needle aspirate and, taking another sample. But the most devastating one, which is actually quite rare, is TB meningitis, and that is the one that actually causes death and long life morbidity. There is some advance recently about looking at CSF, so that's cerebrospinal fluid, looking at novel combinations of biomarkers to look at more rapid diagnosis in CSF. So I think that hopefully will eventually have impact, but I still think, the biggest challenge here is actually the clinical suspicion of having TB meningitis, and what you uniformly see in children who've presented with TB meningitis, which is really a devastating disease. Where I work, it's the most common form of bacterial meningitis in children. is that children have presented many times to healthcare services, on average 4 or 5 times, before someone has thought of TB or TB meningitis. 2/3 of them have a household contact with TB. No one's asked, no one thought of it. So universally, those children have actually had failure to thrive, they've crossed centiles, they're not growing, they're apathetic. So just thinking about TB, especially in a high burden setting, is really important. Because if you don't think about TB, whatever test you use will not actually have the desired impact. But there are some advances, and hopefully those in combination in clinical systems, in clinical algorithms, will hopefully eventually impact a more rapid diagnosis of TB meningitis. Most children who present with that form of disease present very late, so the damage is already done. So the earlier diagnosis is really, really critical.
So we've just got one more online. This is for you, Tony. What could the impact of the technology be on hard-to-reach areas?
Which area?
Hard to reach.
Hard to reach.
Oh, hard to reach. Well, we haven't done the validation yet, but definitely, I think the point of care, and especially for the blood-based detection, especially targeting on the extrapulmonary TB and the HIV-infected patients, I think there will be some impact. Yeah.
Thank you.
Yeah.
And just checking if there's any questions from the room at all. Christoph, I know you.
I would just like to remind everyone that these are highlights from the conference, but there's so much more. We are seeing now biomarkers that are predicting disease in different ways and used as tools for monitoring. We observe digital radiology, where with a little camera put on a pole, you could make pictures. I have a colleague in Kharkiv in Ukraine who showed me just pictures last week, how they went with that little digital imaging or X-ray device into the frontline fields to do TB screening. So this is amazing. And look at the industry booths here that are presenting several of these new tools, but also at the presentations. There's stool-based diagnostics, there's tongue swabs, things that we were not thinking about. Why were we not thinking about this? We could have done these studies 20 years ago, but they were just, the time was not ripe. And we are seeing, apart from that new compound from the TB Alliance, many other compounds that are now being evaluated. We currently have 17 new entities in phase 1 and phase 2 clinical development, and a lot of those are actually here presented in different settings at the Union. There's also one field that we have totally neglected over the past decades, and this is post-tuberculosis lung disease. We thought that when the bacteria are dead, the disease has been overcome, but many of those affected by pulmonary tuberculosis are suffering lifelong, and there's a different degree of destruction that is not being reconstructed by scarring and our immune system, despite getting rid of the bacteria, and that's becoming a very big topic currently in the TB field in both children and in adults. And not so much yet, but next year in Bali, hopefully we will see more of non-tuberculous mycobacteria infections, so stay tuned. Try already as soon as possible as the announcement comes to sign up, and stay with us at the Union. Thank you very much.
Thank you all for attending in the room and online. Again, tomorrow, same room, same time. Let me or Charlie know if you've got any further questions. We're happy to set up further interviews, media huddles. Just get in touch. Thank you all.
Thanks for being here.
Pleasure to meet you.
Oh,
Wonderful.
yeah,
Wonderful.
thank you. Can I take a picture for your website and make sure, because we also like to know how those biomarkers. cells should function and kill the bacteria, but it's actually not. And all those stuff like CD4 cells, CD4 cells, they function, and all the inflammatory gene expression goes twice. So yeah, we keep this. Picture of your pack and make sure I send it to you. Oh yeah. And next time Really? Are you going?
Oh, no, it's fine. I'm happy to chat. Yeah. Yeah. You just let me know.
Oh, okay. Then organize this. And spend 2 days.
Yeah, great. Actually, we're going for next. It's really easy.
Spending 2 days. Yeah, because last time.
Maybe once a month. Yeah. I'm pregnant for that. It's so warm, yeah. Yeah, it's.
I'll just send you the list right away.
Yeah, go for it. That's a Saturday. So you're gonna attend?
Oh, no.
I am going. I'm going this Sunday, and I'm meeting with the Minister of Health that week. So I'm excited for that meeting.
Yeah.
Yeah, so we're from where I'm going. Yeah, I just meant to get there. So I still check the ones away from the country. I'm in Georgia.
I looked you up last night.
Yeah,
I have this problem where people don't know if I'm male or female, so I looked you up too.
Oh, no, I
looked you up too now.
So, do you remember Esther?
Do you do she, her? I don't do that.
Now I do. Like, because I was, I wrote a book chapter with this researcher.
And you never met?
And I knew her for years. And she couldn't get an email. And then one time we met via Zoom. And then she was like, oh, I thought you were a man this whole time. I'm like, Esther, we wrote a book together. That was Esther. I was like, you have Googled me.
So I Googled you last night.
In India, Joy is a boy's name.
Really?
It's a girl's name. Yeah, there is Kerala and Bengal are the states where Joy is. So whenever they call out Joy, they look at the boys like
Hi.
Hi, Gregorio. Nice to meet you. Nice to see you in person. Thank you for coming.
Thank you very much.
Thank you all for coming, seriously. This is great.
Hi, Joy. Nice.
I'm sorry for sending presentations so late yesterday.
Oh, no worries. I will put, don't worry about it at all. Thank you for.
Yeah, that's what
Okay, awesome. And I think if it's there, then they will do the time integrated.
That's not ours, that.
No.
Like the time. So some books have that integrated. Like there's like a bar at the top with like green and it gets red when you're close to. But some of them don't. I don't know if this one has it.
I'm going to my phone.
upstairs.
But Joy said that she will do the time in case there's not. Okay. Well, that way we can have the time question.
Mine is. I sent it to you. Should be 15 to 13 or something.
Okay, that's
Also, when I have it,
Can you say that?
Same.
I'll get through it. I can get through my 15 minutes.
So many emails.
Yeah. Yes, me too. It's so hard for me. It's always that. Well, we were on so fast, but I brought that like the Arabic stuff to English, and but sometimes because I have a different accent, it's like so hard to like say to know what I'm saying, so it's confusing.
Oh, we had a very
Your address. I forward you my presentation.
Okay, so this very nice member of parliament in the Netherlands, and we worked very closely as a yes, it was very involved, and it was a really good job. Actually,
First
or it's
slide
like
is just
something for all and so we worked closely together, and so I asked him to write my like end of year, how did I do, whatever. Yes, and consistently for like 3 years he wrote, she enunciates better than anyone I've ever met. Like, I speak very clearly, but she speaks as if she's on like fast forward.
Like 1.5. That's how I listen.